Our next talk is going to be from Alexandra Lushnikova from Örebro University in Sweden, who will be discussing altered levels of immune checkpoint molecules in colon biopsies and sera from microscopic colitis and ulcerative colitis patients compared to controls. Go ahead when you feel ready, Alexandra, and if people have any questions, feel free to add those through the Q&A.
Yeah, thank you. Just a minute while I try to share my screen. There. Can everyone see my screen?
Yeah, that's great. Go ahead.
That's great. Thanks. So yeah, my name is Alexandra, and I'm a final year medical student at Örebro University in Sweden. And the title of my
myself into slide mode.
Yeah, do you want to go full screen with those slides?
Oh, sorry. Let me try sharing again. There, is that better?
Yeah, that's perfect. Thanks very much.
Great. Sorry about that. And the title of my talk is Altered Levels of Both Inhibitory and Stimulatory Biomarkers in Colon Biopsies and Sera from Microscopic Colitis and Ulcerative Colitis Patients Compared to Controls. So I'd like to start off just by giving a quick introduction to microscopic colitis and the other two forms of more classical inflammatory bowel disease, which are Crohn's disease and ulcerative colitis. So microscopic colitis is an inflammatory bowel condition that commonly affects older individuals and mostly females. The cause is still unknown, and the most common presenting symptom is chronic watery non-bloody diarrhea. And when we look at the mucosa of patients with microscopic colitis, no significant changes can be seen. However, when we look under the microscope, it is then that we can see that there are in fact two subtypes. We have lymphocytic colitis, in which there's an increase in the number of intraepithelial lymphocytes. And there's also collagenous colitis, in which the subepithelial collagen layer is thickened. So the two classical forms of inflammatory bowel disease, these patients have an increased risk of getting colorectal cancer. Yet the opposite has been seen in patients with microscopic colitis. So patients with microscopic colitis actually have a decreased risk of getting colorectal cancer, not only compared to the other patients with the other two classical forms of inflammatory bowel disease, but also decreased risk even compared to the general population. So a major reason why we wanted to do this study was to further examine the immune response in microscopic colitis patients. And this is why we looked at, among other molecules, immune checkpoints, which are the regulators of the immune system. And previous studies have also shown that in microscopic colitis, it is the CD8 positive T cells that are dominant, whereas in Crohn's disease and ulcerative colitis, the CD4 positive T cells are the ones that dominate in the immune response. So our hypothesis was that in microscopic colitis, the immune response is more geared towards immune surveillance than in the other two types of inflammatory bowel disease. And these responses instead contribute to inflammation-associated colorectal cancer. So the method that was used in the study is called Luminex, which is a multiplex immunoassay. And we used a ready-made kit that included 14 immunomodulatory molecules. And here in this table, I've tried to group them according to their function in their membrane-bound form. So here we have some examples of inhibitory molecules, such as EDO, CTLA4, PD-1, and also some stimulatory molecules, such as 4-1BB. And to the right here, we have two examples of molecules that can act either in an inhibitory way or a stimulatory way, depending on which receptor they bind to. So the materials used, we looked at protein lysates that were isolated from colon biopsies, and there were 62 patients included in this group. And the biopsies themselves were collected between 2008 and 2015. And in the serum group, we had 89 samples from 74 patients, as we also looked at 15 patients with collagenous colitis, both before and during treatment. And these samples were collected between 2006 and 2020. So if we now take a quick look at the patient demographics in the biopsies group, unfortunately, this table is still incomplete, as due to some COVID regulations, I wasn't able to go into the hospital and look up all the data. So the age is missing here, but we can see that two groups of controls were included in the study. Both groups were non-inflamed controls, and these are the normal controls that were included, and a group of diarrhea controls. And then we also looked at patients with active disease in both MC subgroups and those patients in histological remission. So we can see a trend towards, there was a higher population of females in the whole patient population. And this makes sense as microscopic colitis is more commonly seen in females. So if we now look at the demographics for the serum samples, all groups had a similar median age at biopsy, around 60 years of age, apart from the 2 ulcerative colitis patients. And we can also see again that the patient population is predominantly female. So moving on to the results, here we have 3 examples of more interesting graphs where the differences can most clearly be seen between the patients with microscopic colitis compared to controls. So if we start by looking at 41BB, which is involved in co-stimulation of T cells, and we saw an increase in 41BB in patients with active collagenous colitis, as well as patients with active ulcerative colitis compared to controls. And if we now focus on EDO, which is an inhibitory immune checkpoint that's involved in immune activation. So if EDO is upregulated, that means that the body's immune response is trying to dampen the inflammation. So we saw an increase in EDO in both subgroups of microscopic colitis and in ulcerative colitis patients with active disease compared to controls. And lastly, here on the right, we have CD28, which is a stimulatory checkpoint. And there we saw an increase in collagenous colitis active patients compared to diarrhea controls. So now we can take a closer look still at the results from the protein lysates in the microscopic colitis group. So as I already mentioned, EDO was increased in both subtypes compared to controls. And in collagenous colitis alone compared to controls, here were the molecules that we saw an increase in. And focusing on the ulcerative colitis patients, we saw an increase in these molecules listed here in comparison to controls. So in both ulcerative colitis and microscopic colitis, we saw significantly increased concentrations of both inhibitory and stimulatory biomarkers. Now, if we now focus on the serum results, we saw essentially the opposite of what we saw in the protein lysates. So here I've included 3 examples of the same molecules that I included on the previous slides. So for 1BB, we saw a significant decrease in patients with active collagenous colitis compared to controls. And in EDO, we also saw a decrease in collagenous colitis patients compared to controls. Whereas for CD28, we saw no significant differences. And in fact, a lot of the samples were below the detection limit. And here is a more detailed summary of the results we saw in the serum. So in both subtypes, levels of TIM3 and CD27 were decreased in comparison to controls. In lymphocytic colitis, these molecules were decreased compared to controls. And lastly, in collagenous colitis patients in remission compared to controls, the levels of these molecules were decreased. So in summary, in the protein lysate samples, we saw increased levels of both inhibitory and stimulatory biomarkers in patients with active microscopic colitis and ulcerative colitis when compared to controls. And interestingly, in some cases, in the cases of these 5 biomarkers, the levels were more increased in collagenous colitis than in ulcerative colitis, despite the presence of more subtle inflammation in collagenous colitis, which cannot even be seen microscopically. And the general trend that we also noted was that the levels of these biomarkers were decreased in patients in remission compared to those patients in active disease. And one limitation of our study was that when we looked in the protein lysates, we do not know whether we are measuring the soluble or membrane-bound forms of these checkpoints and other molecules. And in the serum, we saw decreased levels of both inhibitory and stimulatory biomarkers in patients with microscopic colitis compared to controls. So that brings me to the end of my presentation, and thank you very much for giving me this opportunity and for listening. I'm happy to try to answer any questions.
Amazing. Thanks so much. That was an excellent presentation. I'll open up the floor for any questions right now. I don't see anything in the Q&A, but again, once your time is all up here, you'll have that option to continue to see those questions in the Q&A as they come in. Oh, I do see one question right now. Were you able to control in your analysis for different therapies?
That's a very good question, and I'm still in the process of gathering more information about the medications that the patients were on and disease duration and things like that. But what I can say is that in the microscopic colitis group, all patients, the typical therapy there is budesonide, which is a corticosteroid. But in the other patient groups, I do not dare to say whether they were all equal in terms of disease duration and the types of medication they were on.
Great, thanks very much. Dr. Fish or Payam, any questions here?
Yeah, no, that was one of my questions, what the therapies that these patients are obviously on, and notably that the action is all in the tissue itself and not in the circulation. And I wondered whether are there any studies where they're looking at targeting 4-1BB or PD-L1 in these patients? Are you aware of any of these?
Do you mean patients with microscopic colitis or?
Yes.
Yeah. No, that's a really interesting consideration. So for now, the typical treatment is budesonide, and most patients do go into remission after that, but it's a chronic disease with lots of relapses and significantly impacts the quality of life. So I do not know of any studies that have looked at these treatments in microscopic colitis patients, but it would be really interesting to look into that more.
Thank you for a great presentation, for joining us, Alexandra. I had a question, maybe more about other prospective treatments in your opinion, since you're a medical student, and I assume it's something you consider. What are your thoughts about some of the alternative treatments for colitis proposed by people such as Eric Paymer, where you are trying to look at the roles of microbiota and clinical outcomes? So, they're doing fecal transfer experiments and so on. What are your thoughts about the realistic prospects of, these sort of treatments?
That's a really interesting question. I think all that I can say is that I've also seen in the microscopic colitis patients that fecal stream diversion and fecal transplantation has had good effects in a small amount of studies, but the study population is quite small. So in theory, it would be a possible treatment, but yeah, I'm not sure if I can fully answer your question.
Great talk. Thank you.
Thank you.
Great. Thanks very much again. That was a really great presentation. If any other questions come in through the Q&A, I do have another one actually in the Q&A before I let you go. Any idea what immune cells are responsible for these checkpoint molecules in the tissue?
Sorry, could you repeat your question?
Yeah. Any idea what immune cells are responsible for the checkpoint molecules in the tissue?
Okay. That's something that we will have to look into further, and I don't have an answer on the top of my head. But we have seen that there is that the immune response in microscopic colitis is more CD8 positive T cell based. Yeah, I wish I had a better answer.
All good. That's great. Thanks very much. If any other questions come in, those will be in the Q&A.
Yeah.
Great. Thanks again.
Thank you.